Thanks. So I'm going to talk about the development of a streptococcal vaccine candidate that we've been pursuing in partnership with our very good colleagues at U of A, particularly Mike Houghton, Lorne Terrell, and Greg Terrell, and others who I'll refer to as we go on. It's been a team effort. I started working on Strep though back in 1990, but really getting into the clinical trials only 5 years ago. A bit more than that now, actually. But let's talk about the burden of streptococcal disease and why we're looking to make a vaccine to strep. The Strep A organism, Group A Streptococcus, I'll refer to it as Strep A, that's how it's commonly called these days, causes more disease complexes, symptom complexes than any other microorganism. But the two major diseases that it causes are rheumatic heart disease and invasive streptococcal disease. Now, this is the same germ that causes school sores, we call them here in Australia, or impetigo, on kids when they fall over in the playground and scratch their knee, they often get a streptococcal infection. It's the same germ that causes bacterial tonsillitis. Causes many, many things, but they're common ones. But these two diseases are serious. Together, they're responsible for about 500,000 young lives lost every year. Now, in Australia and Indigenous populations in many parts of the world, but particularly in Australia, rheumatic heart disease is a major problem. This is a map of the northern part of Australia. And in this community here, in Maningrida, 1 in 20 children suffer from rheumatic heart disease. That's a very awful statistic. It doesn't reflect well on my country at all, but that is the reality. Rheumatic heart disease is, let's go back a bit. Rheumatic heart disease is one part of a disease complex called rheumatic fever. So rheumatic fever is an autoimmune disease. When you get a streptococcal infection, about 1 or 2% of the population, if it's not treated, 1 or 2% of the population will get or can get rheumatic fever. And it's when the immune system, which has been stimulated by Strep A infection, either in the skin or the throat, turns on the body, turns on the heart, causing rheumatic heart disease and scarring of the heart valves, turns on the brain, causing a disease called Sydenham's chorea. And I'll show a video of that in a moment. Turns on the skin, the joints, and you get arthritis, arthralgia, painful joints. But it's really the heart. All those conditions resolve except the heart. So the heart condition does not resolve, and every time you get a streptococcal infection, after that, it will make it worse. So the only thing you can do is give these kids monthly injections of long-acting penicillin for 15 to 20 years to get them over that period where their body needs to develop immunity to strep naturally. So let's just talk a video of rheumatic fever. So rheumatic heart disease is shocking disease, but the rheumatic fever, the Sydenham's chorea is a disease. I'll show you on the slide here. So it's video here.
Would you mind just shutting the door for us?
I'll just pause it for a second. That little Aboriginal girl, it's quite common, rheumatic fever, quite common, although this video was taken some years ago, as you can see, and you can tell by what the doctor's wearing. It's not modern doctor attire, that's for sure. But this little girl is, her mind's fine in the sense that her thought processes are fine, but she can't control her movements. That's what we're seeing now. And it's due to antibodies induced by strep affecting the nuclei in the brain. Oops, let's go back, sorry.
Cool. Would you mind just shutting the door for us? Good, Josie. That's great. Now, we'll just do this for a few minutes, all right? Thanks for doing this. Now, what I want to do, okay, see how well you can write your name now. And then when we start the medicine, you're going to get to the right name again and see if you can do it better. Okay? Can you try to write your name for us?
I think you get the idea. Josie is unable to do basic things with her body, which are important for her well-being. She can't eat, she can't dress herself, she can't go to the toilet by herself. So this lasts for some months, several months, and then it will resolve as the antibodies wane. But as I said, it's a heart disease that doesn't wane. So Indigenous populations in Canada also suffer serious consequences of streptococcal infection. And although rheumatic fever does occur in First Nations people in Canada, invasive streptococcal disease is more prominent. And this started from Dr. Greg Terrell, one of my good colleagues and friends at U of A, Alberta Health Services, plotting the incidence per 100,000 in First Nations kids in Alberta versus non-First Nations kids. And they have a very high rate. It's 5 to 8 times higher than the general population for invasive strep disease. So invasive strep disease, it's referred to by different names in the media. Necrotizing fasciitis is a more technical term. You can get streptococcal toxic shock. The press often refer to flesh-eating bug disease, but it's got a high mortality. Mortality up to 25%, and it's much more common in First Nations people than non-First Nations people. But it does occur throughout communities around the world. Altogether, about 150,000 people die of that disease every year. So this is the germ responsible for it all. So in invasive disease, the germ is actually in the tissue. In rheumatic heart disease, the germ is not in the heart. You do not find it in the heart. What's happening in the heart is an immune response started by the germ turning on the heart. But the invasive disease, the germ is certainly there, causing toxic shock as well. So we have been working on a vaccine candidate for a number of years. And one of the challenges with strep in terms of vaccine, there were two main challenges. One is there's about 250 known serotypes of the organism defined by this protein called the M protein. And the amino terminus of the M protein is different in every strain, essentially. And so you can develop immunity to one strain, but next time you get a streptococcal infection, it'll be a different strain and you're not immune. So one challenge is how do you overcome that? So our approach to that first challenge was to look at the conserved region right down here, which is highly conserved, unlike this amino-terminal region, highly conserved. And we came across a couple of related peptides down there, which I won't go into too much detail in interest of time. But but antibodies to those peptides can destroy every strain of strep in laboratory animals, at least, that we've looked at so far. And we've looked at about, I think, 8 or 10 different group A strep strains, and we can protect against each one of them. So problem number one is how do we address the strain variation? Problem number two is, well, we don't want a vaccine to actually cause the rheumatic fever or the rheumatic heart disease, which a streptococcal infection can give you. So we have to be very sure that the vaccine doesn't induce a pathogenic response. Turns out that there's a lot of evidence now that the pathogenic response is due to antibody responses to this blue region here, this B repeat region, not to the C repeat region. But nevertheless, that's what the data suggests. It doesn't prove that you can't cause disease by an immune response to any part of the organism. It has to be formally proven and tested. So they're the way we took. So we've looked at this region down here and made as small as epitope as possible to minimize any chance of causing autoimmune pathology. Now, that's the main part of our vaccine, the conserved region of the M protein. What I refer to as the auxiliary vaccine epitope, K4S2, is located on an anti-neutrophil virulence factor called SPICEP. So organisms streptococcal organisms all express SPICEP. The more virulent ones express more SPICEP. What SPICEP does is it blocks neutrophils from coming into the site of infection. So to make a protective immune response using the M protein as a main vaccine antigen, you have antibodies to the M protein, but those antibodies work with neutrophils. And the neutrophils have to come in to basically bind the opsonized bacteria and kill it. So if the neutrophils don't come in, the antibodies to the M protein don't work. And what the organism can do by making this molecule called spicep, it'll destroy the interleukin-8, which is the neutrophil protease, neutrophil chemoattractant. It destroy that neutrophil chemoattractant. They don't come in. So to guard against that, we've made antibodies to. We defined an epitope on spicep called K4S2 and made antibodies to that as part of the vaccine. So antibodies block spicep and antibodies to the M protein opsonize the bacteria. That's the overall goal. So we have 2 vaccines in development at U of A right now in clinical trial. One called J8, conjugated to this molecule called CRIM, which is like it's a carrier protein found in many vaccines, and K4S2. And the other is this related molecule, peptide called P*17, conjugated to CRIM, plus K4S2. So M protein, spicep, M protein, spicep. We're testing those for safety and for immunogenicity. And I'll come back to that data in a moment, but I want to give you some preclinical data first. How are we going for time? A bit slower than I thought. So the vaccine platform we're using in Canada for the human trial is alhydrogel. So we mix these 2 vaccines together, and this is and we add them with alum, which, as you know, is a standard adjuvant for many human vaccines. It's given by 3 intramuscular injections. This is a trial at the North Alberta Clinical Trial Research Centre in the U of A. When we look at that particular vaccine in preclinical studies, what we see extremely good protection in terms of reduction in bacterial load following vaccination. Here we're looking at we vaccinated mice and looked at and challenged them with an organism, and looked at the number of colonies that are growing in the mucosal sites. That's the nasal secretions or the throat swab, or in the organs of the upper respiratory tract or lower respiratory tract. The NOLT, which is a nasal-associated lymphoid tissue or mouse tonsils, blood, spleen, and lungs. And we can see this percent reduction is very significant with this vaccine. This is for the P*17 K4S2 vaccine. We have similar data for the J8 K4S2 vaccine. Now, this immunity is long-lived. So in this particular experiment, the mice were vaccinated and we let them wait 9 months following vaccination, and then we challenged them and we measured the number of colonies of strep in the TS is throat swab, NOLT is the mouse tonsils and the lungs. And we also looked at the clinical scores. That was done blinded, so observer blinded to what groups were given. And you can see the vaccine was highly effective. It's causing close enough to 100% reduction, complete reduction almost, in colony counts in the various tissues, and a significantly improved clinical score following challenge. We find, this is published, that infection will boost the immune response. So you vaccinate mice. This one was J8, and DT is diphtheria toxoid. It's very close related to CRIM. It's cheaper to make, so we use it for research grade experiments often. Vaccinate mice, just a J8 DT. Then we give them these 4 different infections, and we measure the number of J8 specific antibodies, and we see antibody secreting cells, I beg your pardon, and we see they increase as a result of infection. So the vaccine can be boosted by exposure to the organism, which is what we were hoping to see. And that's measuring, that last slide was measuring the number of antibody secreting cells in terms of the reduction in colony counts following a skin infection. In this stage, initially, the mice were showing a 97% reduction in colony counts in the skin. Every time they get an infection, that increases. So we're seeing functional immunological boosting as a result of infection, which the hope is, therefore, if this vaccine is effective in humans as it is in mice, you'll never have to require booster injections, because exposure to organisms in the community will do that for you. Now, what about safety? So this is an important point. So working with my colleague, Nackerman Kathieson at the University of New England, with Madeline Cunningham, he initially developed this very important rat model. It's the only model, it's the only animal model for rheumatic fever. So you give the, you have to basically vaccinate the mice with potent adjuvants, including complete foreign adjuvant. You give them the vaccines. You have to give them pertussis toxin as well. All these things help the immune system and cause inflammation. And then those rats are tested in various ways They have ECGs done on them, believe it or not, or EKGs done on them. They have psychological tests because rheumatic fever causes obsessive compulsive disorder, and we can test that in the rats. And the positive control is if you give the rat recombinant M protein, that's what happens. But we're testing our vaccines. We've done it for J8, P17, and K4S2. I can't show you all the data. But basically here we're looking at the antibodies following immunization of these rats with that adjuvant protocol. And this is looking at antibodies to cardiac myosin in the positive control. And you can see that if you give rats recombinant M protein, they develop antibodies to cardiac myosin, tropomyosin, laminin, tubulin, and dopamine receptors 1 and 2 in the brain. These are all target antigens of rheumatic fever, either rheumatic heart disease or rheumatic chorea. Fortunately, we're very pleased to see that our vaccine did not stimulate antibodies at all to any of these, which is the P*7 and K4S2 here. There you can see all that there. So very pleased by that. This is a carditis score. So this is looking at the inflammation in the heart tissue. It's scored by a veterinary pathologist who looks at and counts the number of inflammatory cells in certain sections of the heart valves and heart muscle. So again, we're very pleased with that. Now that's one platform. I didn't show you the other data about the rats protected against obsessive compulsive disorder. It's fascinating stuff to watch. But they are, and also the EKGs. Little rats can have EKGs set up on their chest, and you can show that there's no prolongation of the PR interval, which you see in rheumatic fever. Fascinating. Anyway, the other vaccine platform is the same antigens, but with a liposome called CAPO1, and we've done experiments with that in mice, and we plan to put that in the clinic hopefully this year. This is showing protection in mice against invasive disease and upper respiratory tract infection following vaccination with the CAPO1 vaccine and CAPO1. Again, % reduction comparison to buffer control. So we're getting 90 to 95% reduction in colony counts. The third platform is we just published this last year. Vicky Osberg was the first author on this paper, and Manisha Pandey was the last author on this paper, and it's a technology which we developed in-house where you make a liposome, and instead of using CRIM as a conjugate to the peptide, you actually embed the peptide in the membrane surface this way. So the peptide is sticking out, and you put diphtheria toxoid inside, unconjugated, but inside the liposome. Then you add a TLR4 agonist called 3D FAD to the vaccine, and it is an amazing platform, because what we see when you vaccinate mice with this is not, we don't have plans for the clinical trial yet, because we'd have to find significant funding to make this for clinical grade. But when you see this particular platform, you see that their IgA persists for over a year, and once you give them another infection, it boosts it. So I mean, I've never seen any adjuvant in mice give you an IgA response that lasts that long. That's the with the FAD. Without the FAD, if you leave the FAD out, you get a good IgA response. Not that good, of course, but it's nowhere near as good if you have the FAD in there. Anyway, so that's a third platform, but that one's not going in the clinic right now. So let's talk about the clinical data. So at NACTRAC, the U of A, we have 5 volunteers who have already received 3 doses of the J8 K4S2 vaccine. We've had 5 volunteers who've already received 3 doses of the P*17 K4S2 vaccine. That's a single blinded study. So we know what they got, but the volunteers didn't know what they got. And we have 5 volunteers who will receive, about to receive the J8 vaccine, another 5 who are about to receive the P*17 vaccine, and 10 volunteers who will receive a Rabivir placebo vaccine. And that's about to start now, and that will be double-blinded, that particular part of the study. So we're halfway through, essentially. So let's just look at some of the antibody levels that we're seeing in the vaccinees. So these are the 5 who got the J8 K4S2 vaccine. Here we're measuring antibodies to J8 which is a peptide. And you can see four of the, and we have a blue, which is what's called T0, that's prior to vaccination. We have the orange square, which is T3, which is 2 weeks after the third vaccination in this particular study. And you can see that 4 of the 5 volunteers have made reasonable antibodies to J8. Very pleased about that. This one here made some little bit of antibody, not much. Now, we waited 6 months, and we've got the 6-month data now after vaccination, and we can see that still the 4 who responded at 2 weeks are responding at 6 months, and so that's very, very good to see. So the level of antibodies are persisting. I didn't actually expect them to persist that well. We know from our animal studies that if you take memory B cells from an immune mouse and put them into a naive mouse which has no antibody to J8, you'll find that those and then challenge those mice are strongly protected. In other words, the memory B cells can be activated so quickly that they can protect those mice. But here we're seeing these mice obviously will have memory B cells, but they've also got persisting antibody made by the plasma cells in the bone marrow, and it's keeping producing it. So that's a very encouraging sign, I think, for going forward. Now, what about the P*17? Let me see what's on the next one. I want to show you now how we actually determine whether antibodies induced by the vaccine, whether they be J8 or P*7 antibodies, how we determine whether those antibodies are actually binding the M protein on the surface of strep. So we want them to do that. It's one thing making antibodies to the vaccine, but they've got to recognize the strep. So we developed this assay where we have a monoclonal antibody happens to be against the P*17 epitope. So we incubate Strep A with serum from the vaccinees, T0 pre-immune or T3 post-immunization. Now, these are human serum, of course, and IVIG is a positive control. We then wash that out. We then probe them with antibody, the mouse antibody to P*17. And the idea is if the T3 serum is bound, there'll be no room left for the P*17 to bind. And then we detect that and we know P*17 binds the M protein. So we detect that by an anti-mouse reagent at a fluorophore. Excuse me. So what we're looking here is we're looking for a shift from right to left in the signal would indicate M protein binding. So here, for example, we're looking at a shift. T0 is the blue, and this is on a log scale, of course. This is the flow plot. And then T0 is, and T3 is the pale green, IVIG is the red. And you can see there is a shift. Now, there's a shift these are the 5 volunteers who were vaccinated with the P*17 K4S2 vaccine. So they've all shifted right to left, and each one, that means as a result of vaccination, these people now have antibodies which are binding the M protein on the surface of group A strep. Now, we're very, very pleased with that, obviously. Question many people have asked me, and no one knows the answer to this: how much antibody do we need to protect? And we honestly can't answer that. These are adult volunteers who are already immune to strep. Adults by the age of 20 or so, some people do get recurrent tonsillitis, I know that, but most people don't. Most people have strep immunity by the age of 20, and these people the volunteers are university students largely, and they will have a good streptococcal immunity. But nevertheless, they've got more antibodies now, which will bind strep than they had previously, and that's the important point. When we looked at we haven't done, this is work in progress very much so. We haven't done all the studies with all the sera yet, because it is some of the assays are quite time consuming. But with the J8 vaccine, we see the same thing. We see a shift from right to left, indicating that that person, as a result of vaccination, had antibodies which now bind the M protein on the surface of strep. Now, let's talk about the other molecule in the vaccine, the K4S2. Now, in this particular trial, we had much less K4S2 than we were hoping for, because we had production issues. So instead of giving them 50 micrograms of K4S2, they only end up getting 6.25 micrograms of K4S2. So not everybody made antibodies to K4S2, unfortunately, but some did. So we were able to actually test whether those antibodies are functional. Now, how do we do that? It's called an IL-8 protection assay. So I mentioned that IL-8 is a chemoattractant for neutrophils. Many of you would know that. But spicet, made by the bacteria, destroys interleukin-8. So the vaccine K4S2 peptide is aimed to induce antibodies which bind and neutralize streptococcal spicet. So the assay is this: you incubate Strep A supernatant as a source of spicet with pre-immune or immune serum. You then add recombinant interleukin-8. You then measure the residual interleukin-8 by ELISA, and you calculate the percent protection of IL-8 relative to positive and negative controls. In a nutshell, that's the assay. It's a fairly straightforward assay, to be honest, but it's a nice assay. It's a functional assay. So these are two volunteers who did make good antibodies to K4S2. So we got here, the blue was T0, so this person already had some antibodies to spicet, or K4S2, as you can see, not surprising. But following vaccination, the antibodies in volunteer number 10 increased. Same over here. Volunteer number 11 had some level of pre-existing immunity. Following vaccination, they went up. Now, how effective were those sera? So what we're seeing here, look at the T0 sera. These are the positive and negative controls here. T0 sera for both patient 10 and patient 11 had even though they had some antibodies, those antibodies were not sufficient to neutralize spicet. Mind you, for the supernatant in this assay, I'll go back here, this supernatant, we used a highly expressing strain of spicet, a highly virulent strain, which makes a lot of spicet. So it may not be surprising that the level of antibodies they had to start with might not have been effective. But when we look at T3 serum, we see that in both of them, patient 10 and patient 11, we are seeing a significant rise in ability to protect IL-8. We're getting now 50+ % protection of IL-8. So that's very encouraging. So we're pleased by that. We would have liked it to have all the volunteers make antibodies to K4S2, but as I said, they only got a very small dose of vaccine. So we're in our next trial coming up, which is going to start hopefully the middle of this year in Australia. We are planning to have a full dose of K4S2 in the vaccine. So these are the upcoming clinical plans. We'll be doing a dose-ranging study in adults, a so-called phase 1. That previous study I just showed you was done of a single dose of vaccine given 3 times, but a single dose. Next one will be a dose-ranging. We'll be testing vaccine efficacy in a human challenge model. So we have colleagues in Melbourne at the Murdoch Children's Research Institute who have the ability to deliberately infect volunteers, adult volunteers in the throat with strep. And we'll be testing our vaccine for efficacy in that model. And then we plan to do a dose-ranging study in young children. This won't be a challenge study. This will be a regular phase 2 study to look at efficacy from community-acquired streptococcal infections. That'll be a study involving over 1,000 children done in Australia. That's our plan moving ahead. We're hoping to start this one here. All the plans are started in the first half of this year, or maybe the middle of the year. The second one probably start in 2025, assuming we can attract sufficient funding to do that. These trials are incredibly expensive. At trial levels, cost of the order of $40 million. They're not cheap to do. So I just want to, that's where the talk comes to an end. I just want to basically thank a number of people, particularly my dear colleagues at U of A, Mike Lawn, Michael Hooks, who was there, is now moved over to Vancouver. Vanessa Mayer Stevenson, who's been running as the clinical doctor running this trial. Alina, Catherine, and Stan Houston, who was involved at the start. And over at Alberta Health Services, Greg Terrell, who's one of the world's foremost streptococcal experts. And a great colleague to work with. I've mentioned my own lab, of course, Manisha, Simone, Vicky, Mayfuse, Aileen. They're the main people. And in the commercial side, we've got Chris and Eloise, who have been critical to this whole program developing moving forward. In Melbourne, I've mentioned our colleagues, Andrew and Josh. Didn't show you any of their data today because we haven't actually done that trial with them yet, but that we have a grant to do that. Our other colleagues, Kathisen at University of New England here, and Rukshan, his research fellow in Armidale in Australia, and colleagues at University of Queensland and Telethon Kids Institute in Perth, and many funding partners over many years. Li Ka-shing certainly gets a big tick here. They've got behind and funded the clinical trial that I showed you the data from, or some of the data as it's emerging from the clinical trial. But many other groups have funded. Obviously, Griffith University, where I work, has been a major supporter of this ever since I've been there, which is 2010. And a number of other groups nationally and internationally. Currently, the Snow Foundation, Olimvax, Australian government through the National Health Medical Research Council, and other groups in the past. And I think that is it. That is it.